The final flash talk of this session is Monisha Singha. Please come up.
Good afternoon, everyone. I'm Monisha Singha, a postdoctoral researcher at IOI University of Oxford. As in this conference, we are talking about AMR problem and different ways of research to tackle AMR. In this talk, I'm going to present how small molecule inhibitors are developed to tackle AMR, with a particular focus on development of broad spectrum inhibitors of MBL enzymes. From a 10-year study in US, it has been found that among all the available antibiotics, the highest proportion of prescribed antibiotics are beta-lactams. Penicillin and cephalosporins, carbapenems, and monobactams are the 4 different class of beta-lactam antibiotics, and they all kill bacteria by interrupting the bacterial cell wall formation. But with time, bacteria has developed resistance towards the beta-lactam antibiotics, and one of the major reasons for this resistance is due to the emergence of 2 types of beta-lactamase enzymes, serine beta-lactamases or SBL and metallo-beta-lactamase or MBL. So SBL hydrolyzes the beta-lactam antibiotic by a nucleophilic serine residue, and fortunately we have a few approved drugs for inhibiting SBL, for example clavulanic acid, which is used in combination with beta-lactam antibiotics like amoxicillin. On the other hand, MBL hydrolyzes beta-lactam ring by a hydroxyl group which is coordinated by 2 zinc metal ions in the active site of MBL. And unfortunately, there are no clinically approved MBL inhibitors so far. So inhibition of MBL is extremely important and yet challenging because MBL hydrolyzes carbapenem antibiotics, which is one of the last resort of antibiotic to treat the serious multi-drug resistant infections. And also, most of the MBL inhibitors target to bind to the zinc metal ions into the active site, which leads to the poor selectivity over human metalloenzymes. Also, due to the structural diversity into the active site of different MBL variants, it is essential to develop broad spectrum MBL inhibitor, which will target multiple clinically relevant MBLs. So this is the drug development pipeline in which every drug molecule has to go through this process to go from laboratory to the market. And here is the taniborbactam, which is both SBL and MBL inhibitor, and it has reached to the phase 3 stage of clinical trial so far. A few years ago, European lead factory performed a high throughput screening, and they have found indole 2-carboxylate 1 as a qualified hit. After that, by Professor Scofield group, the indole carboxylate was further developed to get the molecule EBL2915, which is currently in the lead to candidate stage. The compound EBL2915 showed better inhibition potency against different clinically relevant MBLs in compared to taniborbactam, which can be seen from this PIC50 color chart, where the PIC50 is the negative logarithm of IC50 in molar concentration. So more PIC50 means better inhibition. One of the reason why indole carboxylate was having better activity is because of its mode of inhibition. So rather than just binding to the zinc metal ions in the active site, the indole carboxylate is mimicking the enzyme substrate complex of MBL and carbapenem. So this is the enzyme substrate complex of MBL and carbapenem, and this is the complex of indole carboxylate and MBL. This type of mode of inhibition was also supported by the crystal structure, and this result was published in 2022 in Nature Chemistry Journal. Furthermore, we thought that pyrrole 2-carboxylate, which has a structural similarity with indole 2-carboxylate, can be developed as MBL inhibitor, and it might have the advantage of a better metabolic stability and smaller size. So to develop pyrrole 2-carboxylate as MBL inhibitor, we have attached different alkyl, aryl, and halide groups to to the pyrrole ring of the pyrrole 2-carboxylate, and we had screened more than 25 compounds against the clinically relevant MBL enzymes, VIM1, NDM1, VIM2, and IMP1. And from this PIC50 table, we can see that a phenyl group at R3 position and a methyl group at R2 position helps to have the better activity of pyrrole 2-carboxylate. Although none of these compounds had better activity, or none of these compounds were as good as EBL2915, but further development in the design of pyrrole 2-carboxylate is still under process in our lab. So in conclusion, I have shown that indole 2-carboxylate bind to MBL enzyme by mimicking the enzyme substrate complex, and it has the potential to go for the clinical development in combination with beta-lactam antibiotics. And also I have shown that pyrrole 2-carboxylate can be developed as a drug candidate, and it is hypothesized to have a similar binding mode to that of indole carboxylate. And further structure activity relationship along with the MIC experiment are in progress. I'm thankful to the Professor Scofield Group for my postdoctoral research on MBL inhibitors development, and I'm thankful to the microbiology group of Professor Tim Walsh for the ongoing MIC experiment, and I'm also thankful to IOI for the research funding and giving me this opportunity to present this work. And finally, thank you all for your kind attention.